My name's Karen Joyce from GeoNadir. Also online we have Paul and Joan and many people from around the world as well. I'm joining today from the unceded lands of the Irraganti people up in far north Queensland, otherwise known as Cairns, and really sitting with the Great Barrier Reef as my backyard. What I like to do with these lunch and learn sessions is to give a really short presentation just to sort of set the scene, and then jump in and do the scary thing of live demonstration, which, you know, is always a bit of touch and go, but generally works for us. So it's really cool to do that, and then host some questions. So please, if you've got any questions at all, we love to see them in the chat. Pop them in at any stage, and Paul and Joan will moderate those. If they need to answer it as they go, they will. Otherwise, they'll stop me, or we'll do the question and answer at the end as well. So feel free to please get involved in any way that you feel comfortable to do so. So let's jump right in. What I want to talk about is how we get collaborative analysis going and how we take that all the way to insights. And I'm going to do this today really based on a bit of a case study of something that unfolded in the last 6 to 8 weeks or so at GeoNadir with our partners at James Cook University and Macquarie University as well. So I'm going to tell you a little bit of a story about what was going on, and then jump right in and show you how you can use GeoNadir and drone mapping data to do similar sorts of things for yourself in the environments that you're passionate about. So just as a bit of context, today I'm going to talk about coral bleaching on the Great Barrier Reef. And if you're not familiar with coral bleaching or corals at all, that's perfectly fine. Hopefully you can still take away some understanding of the way we can use drone mapping data for environmental analyses. But to set the scene a little bit here, there was a report put out earlier this year by the Australian Institute of Marine Science that showed the vast extent of coral bleaching on the Great Barrier Reef. And we know this was something that was happening globally around this time as well. So sort of around about March time, there was a global coral bleaching event. And this is what happens when sea surface temperature gets a little bit too warm for the corals to be comfortable. And Joan's going to put a link to that particular report in the chat for you, but hopefully you can appreciate from the map that you see on the screen the extent of the Great Barrier Reef off the coast of Queensland and the different colours there. Not so good being a red to green color if you've got color vision impairment, I'm sorry, but the darker colors there are the extreme levels of coral bleaching through to not quite so extreme up in the northern extent of the Great Barrier Reef. This is what coral bleaching looks like if you're underwater. So this photo from Professor Jane Williamson at Macquarie University. And what you can see in this image is these really, really bright fluorescent corals, and it kind of can look quite pretty, but this is actually coral under stress. And one step further from this is a complete loss of color to white, and then overgrowth by algae if the conditions don't get any better. And so we do often see these types of pictures in the news and in the media demonstrating, oh, this is what's happening to the coral. But what we haven't seen quite so much is really the extent and getting a feel for what this looks like across a broad area. And that was until Dr. George Ross or Jez from the CSIRO here in Australia posted some drone mapping data from Lizard Island earlier this year. And so he put this up on GeoNadir. And so what you can see in this image here is all of those corals that you saw in the previous one close up. This is viewing from above, from his drone, and taking that over an area to drone map that region. And I have had quite a few people say to me, oh, wow, what segmentation did you use, or what's the classification? These are the raw data that you're seeing on the screen now. And it is quite phenomenal, the color that you see and the vibrancy of these data. And when we saw these data appear on our global map on GeoNadir, Joan wrote to me and she said, my goodness, have a look at this data set. This is incredible for all the wrong reasons. But I will jump into this actual data soon so you get a bit of an appreciation for what it looks like. But when we saw these data, knowing the region that it's been captured in, which is not far from where I am, we had a field trip planned to that particular region and took the opportunity to say, well, these data have already been made available on GeoNadir. what if we use our next upcoming field trip to remap that area? So the data that you see on the screen was captured in March of this year. And then the team that I worked with from Macquarie University, James Cook University, Newcastle, and University of Newcastle went back out to the reef in June, so about a month ago now, and captured the exact same area. And this is what they saw. Now, it may be a little bit difficult to see from this level of zoom out. We will jump all the way in and have a look at it on the platform. But basically, there's a considerable area. This is the exact same area on the screen here that you see that we've now assessed as having died post-bleaching. So coral can recover after the bleaching. We're looking here largely at those dark areas being coral mortality. And so our question then was, if we've got these data showing the reef during the bleaching and the reef after the bleaching, how can we take that to a quantitative assessment rather than just a pretty picture on the screen? And so what I'll go through in a moment is exactly those steps that we used to calculate what we determined was 97% coral mortality post-bleaching at that particular area on lizard. And so these are some of the publications that came out of it. It was in The Guardian 2 weeks ago and also in The Conversation. And there's also a blog there on GeoNadir and Jones popping in those links for you in the chat so you can read about this in a little bit more depth as well. If you want to afterwards, of course, ask us any questions. But what we'll do now is jump right into those data and see what then transpired as we went through to analyze these data. So you sort of get the top level answer, if you like, in these publications. But what I want to show you is actually how we went about doing that. So I'm going to jump right over to GeoNadir now. And I know not everybody is a member of GeoNadir, and that's perfectly fine. These are the sorts of things that you can still look at without having an account. You can do more things if you have an account. So this is our global map. All the dots on the map represent areas where people have uploaded drone mapping data. And so I'm just going to go into the east coast of Australia over here so you get a bit of a feel for the location and the context where we're going to jump to. So we have Townsville down here. This is where I'm based in Cairns. And this area just north of where I am here. I'm going to keep zooming in. I'm just using the shift key on my keyboard here to zoom into Lizard Island. So this is the area where we're going to jump all the way into. And just to the tip of Lizard Island here, and you'll see that as I do that, the orthomosaics of the drone mapping data that exist in that area start to become available. So what we can see over here on the left-hand side is 5 different data sets. So there's 3 data sets one data set before the bleaching from 2021, and then 2 during the bleaching, and then the after bleaching. So this is what we saw when we first jumped in and had a look at the data set and could see this really broad area of these bright white and colorful corals that I showed you, an enhanced version when I showed you in that PowerPoint slide deck. So that's what we started working with and went to mimic that exact same area to be able to do the after assessment. So I'm going to jump now into a project that I've already created using the data. So this same area here, you'll see the satellite image in the background. And what you can see on top now, so we've got different layers over on the left-hand side. We've got on top the data set that was captured by Joan, who's online this afternoon as well. So she captured these data as our post-bleaching survey. I can turn that off to have a look at the before that was done by Dr. George Roth. And also there's a previous from 2021 that's not quite so clear when we look at it and doesn't quite cover such a large area. So let's jump first of all into the bleached area. So you'll be able to see, as I zoom in all the way, you'll be able to see that you can see all of the individual corals. You can see the different types of corals as well. So some are a little bit smoother on the top, some are more branching. And there's also water over the top. So there is distortion over the top of the corals here, and you can sort of see these blurry events. I can see a fish here though as well. Quite a few fish over here. So really high detailed data. And then we can put the data that was captured in June this year over the top of that. And we can start to look at just qualitative assessments to start with by clicking on one and using our compare tool. So you'll be able to see that you can directly compare one data set to another. It doesn't give the quantitative analysis at this stage. But so that's what we're looking at. And those are the differences that we really wanted to quantify to be able to say, hey, we came up with 97% coral mortality. We could tell that there's a lot of it, but how did we actually do it? So let's just zoom out a little bit. I'm going to go through a couple of operations that we did, and then I'll show you the final output as though here's one I prepared earlier. So I have my data set on top here, and I'm actually going to start with the during bleaching. And what we did was create 3 quadrats of 10 by 10 meters. So these are three polygons here, but you can see the area. So 99.6 meters squared, so not quite 10 by 10, but pretty close to it. And what we wanted to do was to grab 3 areas that were reasonable representation of really high density coral, a little bit less dense, and then low density. And we wanted to calculate the amount of area covered by bleached coral in that region. Our very first attempt, what we actually did was we thought, oh, that's okay. Let's put a dot in the middle of each coral and let's count the colonies. So we started by going like this, and you'll see it appear over on the left-hand side, these dots related to where all the coral colonies were. But we quickly realized that there were hundreds of these, and in many cases, they were sort of sitting on top of each other. And so this wasn't going to be the best way that we analyze those data. So those points that I've done, I'm just actually going to delete those. I'll just use my keyboard to do that. So really quick and easy being able to do that as well. So the next thing that we thought we needed to do was probably to assess area, and we were going to need to use polygons to do that. And so up the top here, I've got the polygon tool. I can choose if I want to look at a rectangle, a circle, or the polygon itself. And I like odd shaped polygons for this sort of stuff. And so then we just went ahead and really just started drawing blobs of bleached coral around this area. I'm just going to turn snapping off, so I don't want things to lock to each other. So it's quite simple. I started just drawing like this. I was doing this with a stylus at the time, so a little bit easier. And I really want stuff that's on the inside of this polygon. And I'm just going around doing this sort of thing, not taking too much care at this stage, but you should get the picture. And I can even sort of get areas that I'm just going to plop them over the top of each other for the moment. And so you should get the picture. I'm just freely tracing around to get those different polygons. Now, at the moment here, it tells me I've got an area of 16.77. It might be a little bit difficult to see on the screen, but hopefully you can sort of appreciate what that is here. So I can see the shape area of each polygon and the total area of 16.77 meters squared. Now that's going to be an overestimate because I've got things that overlap each other. But what was more important to me though was that I didn't have gaps. So what I wanted to do then, once I'd done the entire area, was to use this tool up here, that's called the union, and mush all of those together. So now I'm down to 1 polygon. You'll see that I've broken up those things that are overlapping each other, and we're down to 16.34 meters squared, which is a better representation without those overlaps. The next thing that we did, and just bearing in mind this was over the whole area, was to say, well, I'm now counting areas that are outside the quadrat that we're interested in. So what I really wanted to do was to crop those down. So again, I can use, under here, we've got the intersect tool, but I can also access it through the toolbox here. So same areas, or using the shortcuts on my keyboard as well. I'm going to intersect those two. And then what I can see when I turn these ones off is that I've now chopped it to the actual, right? So I'm now down to 11.06 meters squared of bleached coral in that quadrat. So I could work out what the percentage in that whole quadrat is, right? By deciding, well, I've got a quadrat that's 99.6 meters squared, 11 meters squared of that is my bleached coral. So I did this for these 3 quadrats, and I looked at it of the bleached area and then afterwards of the recovered images. So if we jump over now, I'll quickly show you this one I prepared earlier looks like. So what you're seeing is these yellow ones are when it was bleached, and the blue ones are what was left as live after the bleaching there as well. So then it's a very simple mathematical equation as to what that percentage is. But of course, I'm just one person, and I'm actually not even a coral biologist or a coral ecologist. So you may not want to trust what I say in terms of assessing the coral for bleaching or for being alive afterwards. So we actually had 5 of us doing this. And so what you can do with GeoNadir is to actually share your projects with people, right? So I can share this project here with anyone with the link, or I can share it with you as an individual based on your email, and I can decide how I want you to interact with that. So we had 5 of us who were all editing, and so ended up with a really big project of 5 people's assessment that turn off everybody else's before it, so we're not biased, and ended up with the assessment of 5 independent scientists assessing the bleached and then the live post-bleaching, and then took the average of those values, and that's where we get to 97% bleached. And we were able to do this all at the same time. So I didn't have to email a shapefile around to my colleagues and then they would do something and email back. It was all done live on the platform, which was a really key thing for us to get to make sure that we can really collaborate in real time with our peers and colleagues as well. So with that, hopefully I've just run through super fast what our process was and maybe give you some ideas about how you can look at analyzing data and assessing change both qualitatively and quantitatively. And I'll open it up to the floor for questions. Actually, while you're thinking of questions, I do have one other thing that I forgot to show as well. So because the way we put the data sets up on GeoNadir allows them to be streamed to other platforms as well, Dr. George Roth from CSIRO then took the process data, so the author mosaics, and has created another little interface here. So you'll be able to see on my screen as I move my mouse around, I'm moving on one side and it's appearing on the other. So we can actually track individual colonies as well. So there's a big patch over here. We can see what it looked like on the left-hand side during the bleaching and on the right-hand side in the post imagery. So we can look at individual colonies and how it's turned out, what it looks like, which is just another really cool way of being able to analyze those data qualitatively. So any questions for me in the chat there, Paul?
I haven't got any questions at the moment. I think Joan's sort of on top of most of them at the moment. But yeah, look, if you do have sort of any questions, feel free to put them into the chat. Whilst we're waiting for that, Karen, do you want to just sort of maybe do a quick run through of some of the tools in the toolbox and how we sort of brought those all together to show people?
Yeah, absolutely. So there's a number of different ways that you can access these tools that I've just quickly demonstrated here. So one way that you can access tools is if you know the right combination of layers to select. So for example, any layer that I select, when I click on this one here, you'll see up the top here will change from the name of my project to the compare tool. So we see compares available there, and also the greenness index, which will show me how green my image is. So any layer that you select, you'll be able to see the compare tool. This, because it's a data set as opposed to a vector overlay, also has that greenness index. So that's one way you can access these tools. You can, if I click the raster and I might click a polygon at the same time, so both are selected, then I'll also be able to access the ability to crop my data set or chop it down like a cookie cutter, or look at the volume of that particular area as well. So those are different options. Paul also mentioned that we've also just released the toolbox. So we've really consolidated all of our tools to be able to quickly and easily view them. So if I click on the toolbox, you'll see I've got a number of different categories here and grouped tools in under specific categories. So today I showed you the shape tools, so the ability to turn snapping on and off, which really locks your polygons together, and lines as well, to union, which split the polygons so that we didn't have overlapping areas, and intersect, which chopped. So those ones available, that you'll see that they're grayed out at the moment because I don't have the correct combination of layers to be able to use those at the moment. You can see in our 3D analysis tools, we've got the terrain profile, you can calculate volume, the greenness index, and some export options as well. So lots of different ways that you can access the tools and also have a look at the shortcuts that are there as well that will let you do those. And if you're not sure about any of the tools or how they work, this is really handy. Down the bottom right-hand side of the screen, there's a little AI chatbot, and you can ask questions, and it's actually really good. So you just click on ask a question, and if there's anything that the AI bot doesn't know, then one of us will jump in and answer that question for you as well.
Okay, just quickly, so how are you selecting the multiple layers? You're pressing something on the keyboard. What is it?
Yeah, sure. So for example, if I click the top one and I click Control, I can click the next one, and then those two will be selected. So Control will select things that are not adjacent to each other, or I can click the top one, and if I hold Shift and click the bottom one, then they will all be selected as well. So two different options depending on what the activity is that you want to do. So I could do that and then hit delete on the keyboard, for example, if I wanted to remove anything, and then always Control Z because undo is super important.
Cool. And for those Mac users, it's Command instead of Control.
Yeah.
Alrighty, a question here from Jabez. If we switch to DSM view, will it be easier to trace the corals with the polygon tool?
Yeah, that's a really good question. Let's take a look at it, hey? So let's have a look, particularly in the after image. So it was a much lower tide. Joan can probably speak to this, how low that tide was on the day when she captured the author mosaic. Well, let's have a look. Let's just turn off some of the others. I like this little tool actually, so only show this layer. I'll turn everything else off and just leave this one on rather than clicking each individual off. Let's turn on the DSM and off the ortho. Well, that's not a good DSM though, Joan. What's up with that one? That was not a good demonstration there.
Joan, color yellow?
Yeah. The DSM looking like that.
That could be potentially everything is very low and very little variation. Because with that color ramp, I'm not 100% sure the range that we have on the platform at this moment. That could be one of the reasons.
Yeah, right. So indirect answer to your question there, Habits. No, at the moment you can't see the individual corals, but that's actually something that I will take back to our team because we do have other data sets where you can see a lot more variation, particularly with those super low tide data sets as well. And the other part that we'll see coming in the next few weeks is the ability to change the contrast in your data sets as well. So you'll be able to highlight a really small range. It's always really difficult with underwater stuff though. Yeah, I take your point. Yeah, with forests, it's definitely easier with forests or houses if you want to do the outline of houses and that sort of thing as well.
Cool. If
Any other questions or thoughts?
Yeah, if people have got questions, feel free to put them in the chat or come off mute as well. I know we've got a few people in here that are analysing data and doing different things. So if you've got any questions about anything else on the analysis tools, then feel free to just ask. Karen, applicability of this here to other data sets that are not in the marine environment as well. What are your sort of thoughts?
Yeah, definitely. So for me, a lot of my work is in the marine environment, but not everything is. So I do have other work that's looking either at, say, coastal change or weeds mapping as well. So a variety of different areas. And in many ways, those types of ecosystems or environments are a lot easier to do this type of work in. So the tools are not specific to working on a coral reef at all. You can use them for anything. And in particular, for me, having that ability to be able to collaborate with people has made things really easy. And all I need to do is to drop a link in an email to people or invite them. So I can actually do that now. So I might actually drop this link in the chat here. So this is the link to the project. And if you go into that, I'll just make sure that anyone with the link can comment. So you'll be able to pop in, and as people do drop in, we'll see that little avatar pop up the top here. So you can see Paul's in there. Someone who's not signed in, so they come in as anonymous, will be in there. You'll have a limited number of those tools, so all the tools won't be available to you unless you're editing. So you'll have these ones, and I can see people's mice moving around as well on the screen. So you can tell, oh, I better not be working in that area doing some digitizing, because maybe somebody else might be working in that area as well. But you're able to go in, turn layers on and off, and it won't affect how I see things. But if you are digitizing things, it will affect. So what I can do actually is I might, Paul, I'll just put you in, or Joan, I'll pop you in as an editor, hey? You're already in there. Joan, do you want to pop in and you can do some editing, and we'll see how that pops up on the platform as well.
Cool. While Joan's jumping in there, Karen. So a question from David. What's some current or planned AI to automate some of this workflow? You want to talk through sort of maybe what the automated workflows we have up already are, and then what we're looking at doing next.
Yeah, great question. Thanks, David. So we do a lot of that stuff working in the background to think about how can we make everybody's lives easier to be able to pull out some of this information and understand our own insights as well. So we are working in the back end to look at segmentation, so being able to pull out individual objects and then potentially being able to identify those objects as well. Joan's done some really cool work on identifying certain features from buildings to solar panels to cars and bicycles even. So some of the urban features are a lot easier to do than some of the more natural ecosystem work. Because we now have this work that we've done here labeling through the number of different scientists, the different areas of coral bleaching, we'll be able to start look at how do we push that into a more automated workflow as well to map the whole area. It's definitely not my intention just to stay at these 3 squares that we're looking at. So yeah, really working a lot in that research development space and looking at how we can bring that to you as more automated flows as well. You'll see Joan has been digitizing on the screen as well, so they're popping up automatically as she's doing that. I can see the things that she's drawn. I also have the ability to delete or move or whatever I like. So that's really helpful when we're collaborating in that space to be able to do that real time as well.
Karen, they're just so from someone else there on, are you able to run any image classification processes to at least identify existing dead corals and coral sands for exclusion in the areas of interest to assist with the digitizing? So you spoke a little bit about that, but do you want to talk a bit more?
Yeah, so at the moment, we don't have any of those classification type tools available on GeoNadir, but what you can do, there's two different ways that you can achieve what you're looking at. The first is to stream the data into another platform. So we have a tile mapping service which allows you to pull it straight into ArcGIS, QGIS, or like I showed quickly before what Dr. Roth had done, pulling things over here. Your other alternative is to download the data set. So you can download the DSM, DTM, and OrthoMosaic and do the same sorts of things. So in the GIS or image processing packages that you're used to using, these data sets will all easily be imported or uploaded into those platforms, and you can continue to use any of those workflows that you're used to using. I think one of the biggest challenges for me, and one of the reasons that we've been developing GeoNadir this way, is that there are a lot of tools that are available for satellite data, but none that really do that well for the super high detail of drone data that we have. So we do have a bit of a gap in the knowledge of how to best automate analyzing these data. And that's why it's such an area of big research for us to get to that stage as well.
Cool. A message from Eric, question from Eric. How are the DTMs created when the photos are capturing both earth and water?
Yeah, that's a really good question. I would say that the DTMs are a visualization, and they are not necessarily representing reality. And it really depends on the depth of the water and the clarity of the water as well. So the mathematical equation is that it's looking at areas of lumps and bumps, basically classifying those areas that are higher and lower, and then trying to estimate what things are that are above a baseline, and then it removes those things. So I would absolutely not 100% rely on any form of mathematical model in this way, but that's the theory of the mathematics that goes behind how that's generated. The DTMs also don't work particularly well if you've got really dense forest, for example, because the model still needs to be able to see at some points where the ground is so it can chop off the trees. So in an area where you've only got dense forest and it doesn't have any ground points, that doesn't work that well either. It will still give a result, but I guess it's just something for you as a user to understand that there's a grain of salt with some of these products that get created as well.
Oh, anyone got any other questions? Karen, maybe do you want to just sort of talk a little bit about, so, you know, it was noticed that Frank said he's, oh no, it was, well, there are new tools still exploring. Do you want to sort of talk about, you know, sort of how we're releasing out tools and, you know, we let people know in the emails that they're coming out, but yeah, just sort of what we've got planned. We've just released volume that's coming out. Anything to sort of talk about some of these tools?
Yeah, cool. So I guess just as a general overview for people, we release every 2 weeks, usually at lunchtime on a Monday. We push out a new release. And sometimes that's a big fix on any major bugs that we've had, or maybe making things a little shinier and glossier on the platform. And most times there's also some new feature. So last Monday, we released the ability to calculate volume within a polygon on a data set that you're looking at. 2 weeks before that, you could do a terrain profile, so like a cross-section of your elevation models. And next Monday, we'll see the ability to be able to click on any pixel or anywhere in your map, and it will return what the value of that pixel is or what the feature is that you've clicked on as well. So those are the sorts of things that we're constantly releasing as new things or improving what's already there. Every week to fortnight, we do send out an email. So if you'd like to stay in touch, please make sure that you're on our newsletter. And if you're not receiving it, let us know as well. Sometimes they're not getting through spam filters. And also on socials as well. So LinkedIn, Twitter, and Facebook as well. We're always posting new things and adding new blogs of those new features as well. Mostly, I guess I'd also like to say, if there's something that you'd like to see, shoot us an email or send us a DM through socials and let us know. So we're constantly looking for new ideas that are really going to add value to the people that use our tools. Or if there's something that you're like, oh, this is okay, but it's not convenient in this particular way, or this is a rub point, or this is a bug, whatever, please let us know. And we'll do our best to see how we can improve it for you, either through a new feature or workaround or whatever it might be. But we really love hearing from you.
Well, Karen, do you want to just show quickly the terrain profile and how that one works?
Yes, can do. We'll go into a different project. Yeah, so this is a project that I've been playing with. It's an area where there was a charity sale done at a local school or community center in the US. And so what we can do here is, when we're looking at a terrain profile, it's really trying to understand what the lumps and bumps of the earth are. So if we have a look, this is the digital surface model of this area here. So just for reference, our orthomosaic that I can swipe over like that. So you can see where the lumps and bumps of the trees and the buildings and all the different stalls that they've got there. There's some tents and all sorts of bits and pieces there. If we were to create a line through, say, these buildings here, and so I'm multi-selecting the line and the data set here. You'll see this little terrain profile pop up here, or you could also access it through the toolbox as a 3D analysis. So I can click on that. And what that does is just, it looks at the elevation there, and you can see it now comes up with this cross section. So the orange line here is what its estimate of the ground is, and the darker orange is the estimate of that surface. So I kind of like to think of it as like a drape over the surface of the earth. And this, the DSM has the lumps and bumps of the trees and the buildings included, and the DTM cuts those and gives you the underlying ground underneath that. So we've used this to look at coastal erosion and how things change. We had a cyclone go through here to have a really, really good look at the erosion on a beach profile. It was really helpful to do that as well. But I like these building ones because it gives you a really good indication of what that cross-section looks like, and it's a little bit more easy to understand what it's doing. You can also do with a polygon. So if I put my data set on my polygon, I can see I've got a volume tool here. So as it's going to calculate this, it will look by default at DSM, but we'll change to DTM if we want to, once it pops up. It'll take a few moments to calculate this one. So you now can see this three-dimensional model here that I'm just interacting with by using my mouse to click and my scroll bar to move around. And it's calculating where a base height would be. At the moment, it's deciding that from the polygon. So these four squares, the four corners that I've got here, going to estimate what the elevation is at that point. And we'll say that's the base height. And then I've got a certain amount below that. So 11 meters cubed below it and 621 meters cubed above that. So we have people using this type of tool for looking at cut fill analysis, looking at piles of dirt, what needs to be removed, what can be pushed in, that sort of thing. You can change from DSM to DTM. You can also change to decide if you want the average of the polygon there instead of the lowest, which is the default. So a couple of different options there as well. Totally different to coral bleaching, but yeah, different tools that you can play with.
Cool. We haven't got too any sort of other questions. Just a comment from Frank that he's done some training shortly around LiDAR surveys. Do you want to just sort of talk a little bit about LiDAR and when that there is best used versus when that there is maybe not best used? We'll dig into a few questions around whether we do process LiDAR data.
Yeah, so we're not processing LiDAR data at the moment. LiDAR data is great in areas where there's quite a bit of vegetation. If you've got sparse vegetation, LiDAR is not necessary. So LiDAR is really good for getting through the canopy of trees to give you the baseline elevation. So when we're looking at the DTM here, LiDAR will give a better result of DTM if there's trees that need to go through. Downside of LiDAR is it's expensive, it weighs more, so you typically need a larger drone, which puts you into different licensing categories. And the processing is a lot harder, has a steeper learning curve, and requires heavier computing power and then more storage as well. So there's some use cases where it's really, really important, but a lot of the time, photogrammetry using a straight RGB or red, green, blue camera with a commercial drone is going to do the job for most use cases.
Cool. All right. Look, I think we haven't got any other questions in the chat. Anything final to add there, Karen?
No, I don't think so. Just please, if you've got questions and you feel like asking them, but not in a more public forum, please let us know. Reach out to us on email or socials or as you like. The questions, comments, we're always happy to hear them. And thanks so much for joining today.